We go on with the next speaker, which is Fabiolinero Fabrin. So, Fabiolinero Fabrin is a research fellow at the National Enterprise for Nanoscience and Nanotechnology, NEST, in Pisa. He graduated in Materials Nanotechnology at the University of Pisa, where he also started working on piezoelectric electrospun nanofibers. His research is focused on the development of fibrous coatings and the study of how the surface morphology can affect the interactions with the external environment. The presentation is entitled Antifouling Properties of Electrospun Polymer Coatings Induced by Surface Morphology. Please, Fabio.
Thank you for your presentation, and thank you everyone to be here this Saturday morning. So this morning I will talk about antifouling properties of electrospun polymer coatings induced by surface morphology. In other words, how the structure or the pattern of the surface can affect the interaction between the surface and the external environment. Okay. So, like every research or topic that we have heard these days, there is a fundamental problem that we have to deal with. In our case, we have observed that bacterial colonization of implanted biomedical devices is one of the main causes of healthcare-associated infections. And it is estimated in Europe a number of 8.9 million combined in the European hospitals and long-term care facilities. To give another perspective, on a given day, we are talking about a number of 1/26 patients in long-term facilities and 1/15 patients in hospitals that have these healthcare-associated infections. Of course, there are different ways to prevent these problems. One of these is like training the staff, so raising the awareness of this problem, and the other also is increasing the hygiene or isolating infected patients. But when we have to combat, not to deal with, but when we encounter this problem, the main treatments are still based on the use of antibiotics. For sure, we have already known, or we have heard, that the mutation of bacteria is leading the bacteria to become antibiotic resistant. So the European Center for Disease Prevention and Control already estimated that 1/3 of bacteria is resistant to antibiotics. Alternative approaches have been developed, and one of them is, for example, physical antifouling repulsion, that consists in providing surfaces with unfavorable features or barriers for bacterial adhesion, without using any chemicals, so in the most sustainable way possible. Already this morphology of this antifouling repulsion has been seen in nature, and has been seen and studied in nature, and many scientists many times these antifouling, these properties has been seen in nature, and we are talking about super-hydrophobic or self-cleaning surfaces. A way among many nanofabrication techniques, electrospinning is for sure one of the cheapest and upscalable techniques that can produce nanomaterials with super-hydrophobic properties. So in this view, in this context, we tried to fabricate with electrospinning micro nanostructures that show intrinsic antifouling physical repulsion based on the geometries of the fiber meshes. So at first, we produced what we call first-generation coatings that consisted in up to 4 layers of aligned PVDF TRFE, so a piezoelectric polymer, a copolymer of PVDF. So we have produced these coatings up to 4 layers of aligned fibers with alternating orientations with 45 degrees, 60 degrees, or 90 degrees. To collect different layers, we have adopted what we have called a number layer step procedure that consisted simply in rotating the substrate, in this case an aluminum foil, by a specific angle after collecting a layer of fibers. For better reproducibility and aiming to produce coatings with more than 4 layers, we focus our research in the 90 degrees orientation. So we are talking about fibers of different layers that are perpendicular to each other. Here I report a morphological characterization for the first generation samples. We have a mean diameter of around 1 micrometer, and we see that we successfully produce a super-hydrophobic coating with a water contact angle of 140 degrees. To evaluate the antifouling properties, we performed a bacterial adhesion assay with the Staphylococcus, considering the Staphylococcus epidermidis bacteria. The SEM images suggested that the bacteria adhere on the top of the surface and penetrate in certain points of the coating. And the numerical results, so considering the colony forming units per surface unit, we have found that the coating with fibers reduced the bacterial adhesion with respect to surfaces, to the control surfaces without the fibers, but no statistical difference was found between the surface with the coating and without the coating. So the reliability of these results was influenced by different factors. First of all, the samples were very delicate and the fibers were not properly anchored to the substrate. Furthermore, we use a type of bacteria that didn't possess the same dimensions of the same order of the geometrical features of the coating. So, considering such outcomes, we developed what we have called second-generation coatings. So, with the same electrospinning setup, but this time using a plain PVDF TRF film as substrate. In this way, the fibers were more anchored, and we were able to produce coatings up to 10 layers of aligned fibers. Here I reported the water contact angle of the plain PVDF TRF film, in the view then of correlating this water contact angle with the contact angle of the coating. And we can see that the fibers had a larger diameter with respect to the first generation coating, and also in this way we produce a super-hydrophobic coating with a contact angle of 127 degrees. Similarly as before, also here we performed the bacterial adhesion assay, this time considering the Escherichia coli as bacteria, since the matrix features had dimensions of the same order of this type of bacteria. Also this time we can see that the bacteria adhere at the top of the surface, and we did not observe penetration through the surface. The numerical results confirmed what we have seen by the SEM images. So there is a reduction with the coating, with the coating, and there is also a statistical difference. And we have calculated that the reduction of bacterial adhesion was about 69%. So, following these outcomes, we have seen that there is a correlation between geometrical features of the coating and the dimensions of microorganisms. But to fully understand and to fully correlate also the bacterial adhesion assay with the wettability results, we use the Casibacter model. So without going into details, that is also beyond the purpose of this presentation, the Casibacter model is a model used to describe heterogeneous surfaces, and to use this model, we have to measure what is called the fraction of solid and the fraction of air, so the porosity of the surface. To evaluate the effective porosity, we design an image-based approach based on GIMP and ImageAce graphic software. We aimed at measuring the different possible porosities seen by the liquid drop on the surface by assuming different possible penetration levels. After measuring the porosity, so of the 4 outermost layers, as I reported in this image, we measure with the Casibacter model the Casibacter contact angle equilibrium, the Casibacter contact angle at equilibrium. So, for example, if we consider the first generation coating and if we consider the porosity given by the first layer, we calculated a contact angle around 150. If we consider 4 layers, the porosity given by 4 layers in the first generation coating, the theoretical model gives a contact angle of around 120. So, if we consider the previous graph and outline in red the experimental water contact angle measures for the first generation and the second generation coatings, the data suggests a possible correlation. For the second generation coating, so the coating with 10 layers of fibers, the experimental water contact angle was 140 and 127 degrees and is in a good agreement with the contact angle measured by the Casibacter model, considering only the first layer of fibers. On the other hand, if we consider the first generation coating, so the samples with 4 layers, we can see that the experimental result is compatible with the contact angle considering 2 layers of coating. This observation suggested that the drop fell in the Casibacter regime at different layers for each sample. Concerning the second generation, we can see that the result indicated that the drop enters in the Casibacter regime in the first layer of the coating. On the other hand, for the first generation coating, the drop fell in the Casibacter regime in the second layer of the coating. And this is in line in what we have seen with the biological assessment, where for the second generation, the drop and the bacteria stops on the top of the layer, and for the first generation, the drop and the bacteria penetrates in the coating. We also estimated the porosities and then the wettability also with a statistical model, and not only with an imaging procedure that can be biased by the user. To conclude, we have seen how tuning the morphology of the surface can affect its antifouling properties, and precisely by fabricating a multi-layer fibrous coating that raised the wettability of the surface to a hydrophobic level, we have observed that the fact that we have entered in the Casibacter regime, so in a super-hydrophobic regime, and the fact that the surface has geometrical features with dimension comparable to the microorganisms affect substantially the antifouling properties of the surface. And these observations can be used in futures when tailored or to design new surfaces. Furthermore, we have developed an image and statistical procedure to evaluate the porosity and then the future wettability of electrospun meshes. Thanks for your attention.
Are there any questions for Fabio?
Thank you, Fabio. This is Ignacio Solaverrieta from the University of Alicante.
Hi.
I have a little question regarding the electrospinning operation conditions. I would like to ask you if you could tell more or less which was the rotation speed of the collector to obtain those beautiful patterns of aligned fibers.
Yes, to obtain such aligned fibers, we have used 4,500 RPM, so a really high rotational speed. And the electrospinning procedure was about, we collected for 30 seconds. So for a precise time, and we really high rotation of the rotating collector. Otherwise, the fiber would not be that aligned.
Okay, thank you.
Thank you. Fabio, for your presentation and very important subjects of investigation because of the antibiotic resistance. I know that these might be a nasty questions, but just to consider that, I mean, if you study, I've seen you have a very high inoculum of bacteria, 3×10⁴ colony forming units for mils of your suspension. That it's a lot. So my question is, it is comparable what you found in a real hospital environment? And the other point is that the density of the inoculum, it's at the base of the regulation of the mechanism of biofilm formation, because bacteria behave in response to very critical mechanisms of regulation related to the quorum sensing. The quorum sensing is related to the density of the population of the microbial community. So I don't know which are the principles to verify or to assess the formation of biofilm in relation to the inoculum of the bacteria, if you consider this factor, and if the density of the inoculum that you use is similar or comparable to what you can find in a real situation.
So I will be honest, I didn't...
Yeah, I know that it's quite nasty and it's not your...
No, don't worry. It's a good question. No, thank you.
Because the density is regulating the behavior of the community. And if you have a lower density, it might be that they do not form any biofilm because the density is too low. Or I don't know. I mean, I don't know. It depends on the species or the microorganism.
Yes.
But that inoculum is very high. 10⁴ is really high, and I think it's too high for a real situation, at least in an hospital.
No, for sure. I will be honest. I help for the bacterial adhesion assay, but the concentration were decided by other colleagues. So
try to ask them.
Yes, for sure. Really, thank you for your question.
Very important question.
We have a question from Zoom. So can we answer just one question? Excuse me. Why did you use a polymer with piezoelectric properties? So that's a good question also. We used the PVDF TRFE, so the copolymer of PVDF, first of all, because it's chemical stable, and for its structural properties, it's very used in biomedical applications. But what I want to highlight here is that we do not use this material our aim was not to focus on the material, but on the geometrical features and how the geometrical features can affect the interaction. So also practically, we have used this material because the electrospinning setup was optimized for this material. Okay, thank you.
Thank you for an interesting presentation.
Thank you.
My question is, have you considered to introduce in the polymer solution in any antibacterial product? I mean, you have been concentrated on the geometry. Yes. Or the coating. But
what do you mean? Sorry, what do you mean by that? Because
it is possible to introduce in the polymer solution active components. It can be different type of molecules. There is a lot of literature on this.
So if we can introduce the polymer solution with antibacterial components, like? Well, for sure we can. For sure, I think that we have to optimize the solution, and changing the solution with other additives means that we have to change the electrospinning parameters, but for sure we can do it if the additives are soluble in the solvent that we have used. Yes, for sure.
Sorry, I have no voice enough for problem of throat. But we made an electrospinning into the gel, the solution. We would send antibacterial and anti-inflammatory compounds. Because the problem is when you go in the hospital, for example, in the barn, you have to solve the problem quickly because, for example, now you have to put on the skin for 3 days. The plastic surgery change. Each 3 days you have a cost of surgery, and a cost of the product, and the patient has to go in the hospital more times. So we found by electrospinning the possibility to produce a tissue that remain in contact with burns for a week, and so the cost is below, and this is very important, the cost. So not only the product, it's important the kind of tissue, kind of active compound, and the final cost of the product, because if it's too much cost,
yes, for sure,
don't go in the market.
Yes, for sure, it's true.
Thank you.
Thank you.
Okay, thank you, Fabio.
Thank you.